Now we move to our next speaker, Professor Mousa, Endowed Chair, Tenure Professor of Pharmacology, Executive Vice President and Chairman and the Pharmacological Research Institute, New York, USA. He is going to give a talk about impact of nanobiotechnologies in medicine.
Thanks for the invitation. Really thank Dr. Amal Bishlawi for this very successful conference, as usual. And I'd like to thank the chair and the audience for, this is an exciting conference. The future is very bright. You heard about gene editing, gene addition, gene therapy. So there is a lot of technology. And I can tell you this technology comes together to give the best to the patient. With regard to nanotechnology, this nanotechnology has a tremendous promise in conjunction with biotechnology and genomic information, and not in isolation. And with nanotechnology, we can deliver the target into the site of action without minimizing this off-target side effects. So this is the promise. I'm going to show you a lot of case studies very quickly. This is a large, big talk, and so actually I'm going to start the first slides here to show the topic that I will highlight to you, and I like to emphasize this is a nanotechnology and merging with biotechnology in order to approach the future of medicine, which is precision medicine or personalized medicine. The key message that I like to highlight with you is I'm going to take you from the bedside to the bench. I'm going to take you back into the bedside. So I actually move quickly from the bedside to the bench and back from the bench to the bedside. And there is a tremendous economic and social impact of this emerging technology. And nanotechnology is a catalyst for many other technologies, bioinformatics, artificial intelligence, supercomputing, big data. Big data is so we're gonna do things in much smarter way, and the precision and accuracy of diagnosis and diagnostic modalities will be highly enhanced, so we can have a better management of the patient. And nanotechnology in conjunction with biotechnology can help us deliver the future of medicine and its personalization. The key fundamental here too, I live in the US for almost most of my life now. I'm from Egypt, proud to be an Egyptian. The academic industry partnership and the collaboration between academia and people collaborating within the same institute and outside the institute is very key fundamental for success in research and development. I like to go back to history, medicine, and I started actually here in Egypt. There are many highly innovative medicine, digitalis, digoxin. And digoxin was first discovered by ancient Egyptian. This is where history started. Many other medications started here. And digoxin, I can tell you, I spent 2 decades in big pharma and now almost close to 3 decades in academia. And digoxin could not be replaced. In big pharma, people spend billions of dollars to replace digoxin, and despite the fact of its therapeutic index, but still a very valuable drug. And so this is history started. I hope that actually history will regain and the Egyptian scientists here and for the sake of the patient will bring a significant contribution to the advances in science and medicine. The application of nanotechnology has a wide range of application. I'm going to focus on nanomedicine, drug delivery, in particular active targeting. Not just simply to put stuff in a nano shell and deliver it, but using genomic information to attach to the nano surface and make it deliver to the site of action exactly in a highly precise manner. And early diagnosis, biosensors and many other diagnostic modalities can be developed using nano and biotechnology. Lab on a chip and tissue engineering, healthcare delivery, and many other applications, as you can see going down the list here. So this has a huge economic impact on the future of an academic unit or even a nation. And I come from Albany, New York. Albany, New York is the capital of New York State, and it is the capital of the world when it comes to nanotechnology. They are number one site. That site almost started almost about 15 years ago. Now it's worth $15 billion. The government invested $1 billion, it's $15 billion. So if you look at the economic impact, this is what I call it the fourth revolution, an industrial revolution. The three industrial revolution here took 200 years to realize its potential. Even the semiconductor industry was here in the late 200 years after the industrial revolution. It was reactivated using nanotechnology in the wafer and the nano chips. So, and this can be achieved in 10 to 15 years. So if you put a roadmap, this can be highly achieved. So I'm talking about going back and becoming not only a user of medications, but producer and inventor of these medications. This is very important. So we don't have to scream about the cost of the medication, the cost of biologics, which is becoming very abundant. So this is very important to be a player in that arena. And if we look at the Industrial Revolution, we missed the Industrial Revolution in this part of the world. However, we started the history of a good history, but we sit back home and just keep talking about we invented this, we have the pyramids, all of that, and we did not make a contribution. So that train is train of nanotechnology moving with a high speed train. And if you are in the station, if you don't jump on the train and become part of the movement, you will actually, the train will keep going. So we have to, this is a message to all of our Egypt and the Arab Ummah. Why nanotechnology? What's the big deal about nanotechnology? Nanotechnology, it's about the size of the material that we use to package a pharmaceutical or drugs or gene. Actually genes and all of these biologics are very unstable, peptides, unstable. And if you don't protect it in a nano shell, they will be destroyed. But more importantly, just simply packaging it, if you can have the genomic information to, like a missile, to target deliver it into the site of action without avoiding all of the side effect and the off-target effect, you can have a very efficient strategy. So in our traditional medication, usually you take it, it goes everywhere, it has a systemic distribution, but nano is nano targeting, you can deliver that into the hepatocyte, into the liver, if you want to treat the liver. If you have a virus, you want to destroy the virus, you can develop a molecular signature that's unique for the virus that doesn't exist in the human host cells. And that's very easy to do. Bacteria, for an antimicrobial, so you don't have the side effect on the human host cells. So you can assemble a nano shell, you can put sorry about that, the screen is not in, the laser is not moving forward to the right side here, but you can put an imaging probe, so you can image. At the same time, target deliver, so you can achieve what's called therapeutics. You can do treatment and diagnostics simultaneously at the same time. And that's happening, actually, the clinical trial is ongoing at Yale, at MD Anderson, with many anti-cancer nano-targeted delivery targets. So you can improve the efficacy and the safety at the same time. Traditional medicine, in general, they have limited efficacy, and the efficacy of traditional medicine is highly questioned. And you know the story of Vioxx and many other medications. And if we look at the statistics here, the drugs, the class of drug and diseases, you have, this is the percentage of non-responders because of pharmacogenomic variations. So you can have at least 1/2 to 2/3 of the population are not really responding. Only 1/3 get the maximum benefit of the drug, 1/3 get a mediocre benefit, and the other third get nothing but the side effect. This is actually, it should not happen in this time and age, but this is the standard of care as we speak about it today. And if we look at the safety is being questioned. There is actually if you look at the mortality due to adverse events is much greater than any other disease. If you just look at cardiovascular disease, cancer, if you just look at the statistics of mortality secondary to adverse event due to using drugs, it is very high percentage. With nanotechnology and active targeting, the efficacy can be maximized, personalized, and at the same time the safety can be totally improved. That's exactly what I'm trying to highlight to you today. This enabling technology have to work on coordination, and technology assist technology. So if you look at nanotechnology, it would benefit tremendously from biotechnology. You can have the bioinformatic to enable and to accelerate filtering the data, and then of course discovery and development will happen when you integrate technology. We can benefit from existing pharmacotherapy to make it more safer and more effective. Cell and stem cell therapy can benefit from nanotechnology by creating a topography for that surface, nanoscaffolds that will enhance differentiation of these stem cell into specific target. Now we are learning about many other non-tumorigenic stem cell called Mousa cells. These Mousa cells can be nano-target delivery of modulators that can induce the homing of these endogenous non-tumorigenic stem cells, unlike mesenchymal or other stem cells, and that can actually, if you have a tissue damage, that can be move into the tissue to repair the damage, for neuronal injury, for vascularization, for cardiac damage, for bone repair. So these things can happen actually without injecting stem cell. So you can use your indigenous patient, so you don't use immunosuppressant. If you have to put immune cells from another patient, stem cell from another patient, you have to use immunosuppressant. So there is a lot of advances. I wish I have more time. Each of these pockets we can spend a day on it. I'm very interested in commercialization and moving the concept from the bench to the bedside to the market, to the finish line. And if you want to actually advance concepts, this is the nanotechnology, the biotechnology here, and the genomic, the function, the proteomics. All of these tools are very good tools. The design of the nano, the assembly, and then now you need to look at the application. What application are you going to try to drive? And then you have to look at the commercialization. You have to look at IP protection, because no investor will come and put a lot of money to develop your concept unless your concept is protected. So it can be developed and they can become licensing. Safety is very important. If you have to go through the FDA, you have to pay attention to safety. Do no harm. If your drug is very effective, that's great news for you against the competition. But the FDA will not allow anything to cause harm to the patient. So what is the type of material that we can use? Abundance of this material can be used. We can use polymeric nanoparticles, a polymer. We can use solid lipid nanoparticles. There are a whole host of material. The material is very important, the selection of the material for the safety reason. The size of the particles, sometimes the size smaller is better. Sometimes you have to optimize the size of the nanoparticles. And then you have to look at the surface and you can modify the surface. I strongly believe usually in active targeting, trying to develop an active ligand, high affinity ligand, that can carry that shell loaded with a drug inside the nanoparticle here into the site of action. So I can have minimal to no side effect, and I can have the greatest effectiveness. So the idea of just making a nanoparticle and just to give it there, it is not optimal. This was maybe okay 10 years ago, but now it is about active targeting. So I'm going to actually just highlight here, drug discovery and drug development takes a long time. Dr. Amal Bashlawi highlighted a lot of the JAK2 inhibitors, many fascinating targets. The iron chelators, all of these took a long time to be developed. Can nanotechnology, shorten the time to drug discovery, drug development? Can it also mitigate the risk? Because this is a high risk proposition. The failure is much higher than the success. And the answer is yes. You can actually take an existing drugs like heparin. We converted heparin, low molecular weight heparin, into an oral dosage form. Insulin can be formulated into by keeping it inside the shell. Multiple RX like chemotherapy, you can target it, deliver it. You can actually put an active moiety to deliver it into the tumor, only into the tumor. So there's a lot of imaging. You can do imaging. You can do imaging and therapy at the same time. You monitor the therapy. And you can transition. This is the natural product here. There is an action Nutraceuticals. A list here, digoxin. This is discovered by ancient Egyptians. Warfarin, aspirin, heparin, celebrated 100 years, magnificent molecules. Actually, the only thing that handicapped that molecule is its an anticoagulant activity, which is the tail of that elephant. If you can cut that tail, actually you can get a lot of value out of heparin. Heparin has an anti-inflammatory, it stabilizes the endothelial cells, protect the endothelial cells, anti-complement. It is anti-sickling compound. If you can engineer an aldehyde function to get anti-sickling, anti-adhesive. I'm going to talk about it in my lecture about the future of sickle cell disease management. Taxols, statin, these can actually be transitioned from nutraceutical into pharmaceutical, which they are. And the Taxol has been transitioned from a pharmaceutical into nanopharmaceutical, Abraxane, by making a nanoformulation to make it more soluble. But it's not really actively targeted. So you can reformulate many existing drugs to improve the pharmacokinetic and pharmacodynamics, either nutraceutical, pharmaceutical, or even biopharmaceuticals. For devices and rapid sustained delivery, and also for active targeting, active targeting into the site of action. You can actually overcome the blood-brain barrier by delivering drugs across the blood-brain barrier for glioblastoma or for centrally active drugs. Imaging. And this is closer to you here for iron supplements. VIN4, this is actually in an iron nanoparticles coated with sucralose. So there are a lot of attempts by many groups, including our groups here, to make an oral iron supplements that actually better than the brand name VIN4. Everybody is going after making generic of VIN4. You can have iron folate, iron ascorbate, and a combination of these in a different proportion to get the optimal iron hemostasis. The other one, in terms of iron chelation, the iron chelators are very good. Of course, this is okay to manage the situation as we speak, but there are other better future opportunities. You can have a nanoshell, you can have many of these iron chelators in a multivalent, decorating the shell, the surface. That will increase the positive cooperativity of the chelation and the permeability to get into the iron away from the tissues. Cardiac tissue in particular, because cardiac arrhythmia is leading to all iron overload in the heart. The insult of some of the endocrine glands can be actually by removing efficiently a lot of, you can have this surface loaded with multiple molecules of existing iron chelators. You can put a supplements of folic acid and other zinc and others inside the shell to be released to supplement the loss of some of these supplements. So these are actually a lot of groups, including our groups, are working on these strategies. I don't have time to go over it. This is a separate lecture by itself. And in our nanotechnology devices, we have done in the device side, you can coat a catheter with a silver heparin nanoparticle by etching and full immobilization on the outside of the catheter, inside the catheter, which is being undone in the pediatric and the elder population, which stay for 28 days or 1 month. This will eliminate infection and catheter-induced thrombosis. You look at publication, there is thousands of publications on catheter-mediated infection and thrombosis. It's a local problem, it can be solved locally. And instead of actually getting the patient to develop infection or thrombosis, and now you have to give heparin or you have to give an antimicrobial. Medicated contact lenses to deliver drug to the back of the eye, to the unsustained release. And these are nano-based, drug-loaded biodegradable stent. Instead of the metal stent, you can have it biodegradable. Delivering the drugs for 3 to 6 months. And then it will be, eventually it will degrade. You don't have to go back on hemostatic devices that you can actually place or spray on the wound, severe wound, to prevent hemorrhage, severe arterial hemorrhage, at the same time to prevent infection and accelerate wound healing. In the lipid lowering, you heard about atherosclerosis, one of the enzymes. This enzyme here is called PCSK9. This enzyme degrades the LDLR receptor on the liver, in the hepatocyte. And if you degrade that receptor, cholesterol will not have a chance to cycle back to the liver to be catabolized. Familial hypercholesterolemia, this enzyme is highly upregulated. And if now there are FDA-approved antibodies, 2 antibodies, one from Amgen, one from Regeneron. You take it 1 injection every 3 weeks. It will shut down the enzyme. Now you can the receptor is being preserved, and now you can cycle the cholesterol. You can bring cholesterol from a familial hypercholesterolemia or normal patient population who are not resistant or cannot tolerate statins. This is a very effective therapy. So what we did, this is over the past 8 years, we worked with a group to develop an oral small molecule compound and targeted to the hepatocyte, to the enzyme. So we developed that drug, and that drug is about to enter phase 1 clinical trials. It's called B21. That lower LDL cholesterol, you can look at the lowering of LDL cholesterol, and you lower the enzyme simultaneously after oral administration. The only FDA approved 2 antibodies. Everybody is shying away from making a tablet. So this is actually, we think that this is going to be a good replacement for these proof of the concept with the monoclonal antibodies. If you look at the antibodies here, FDA approved to reduce LDL cholesterol, to reduce cardiovascular events, the LDL cholesterol lowering is exactly the same with the antibody. And this is very inexpensive, solid dosage form. The drug will go into the hepatocytes and will lower the enzyme without systemic distribution elsewhere. So this is just an example of active targeting. I do have about 2 minutes left for me. We usually use a lot of molecular genomic information to come up with a targeting moiety that's unique to say the cancer cell or the microenvironment of the cancer cells, cancer stem cells, new vascularization around the cancer cells. And we have a bunch of chemists, high affinity probes that will ligate that receptors and go into the site of action. We can assemble a nano shell, and we can put drug inside like chemotherapy, and even the nano shell with the active targeting is very functional by itself. We can do imaging, PET imaging, or MRI imaging by putting a probe, gadolinium, on the ligands, or we can deliver chemotherapy exquisitely into the site of action to destroy the tumor. And if you discontinue the treatment, the tumor will never come back. It becomes a dead piece of tissue. We have done a lot of histopathology, we published extensively. That concept is actually moving forward to clinical trial at MD Anderson, December 2019, in GBM, pancreatic cancer, and acute leukemia. And again, we can talk about that later. So the era of personalized medicine is coming. By 2030, many of the medications, eventually you can have your name written on the capsules or the tablets. That's going to happen. So the idea about one medicine, one size fits all, is going to disappear, which is not the right way. So you're going to see a lot of rise in the biologics as we witness. Nanotechnology on the rise, orphan designation and orphan indication for a small size population is going to be on the rise. And nanotechnology is going to be on the rise. I said it's being adopted. It's entering the phase of mass production and manufacturing is going to actually become a reality in 2030. Many clinical trials are ongoing now with the nanotechnology. So this is what the future holds. You're going to see multifunctional diagnostic imaging modalities, functional imaging, not anatomical imaging. Many of the imaging we have so far anatomical, to give you anatomical information. We are going to improve efficacy and safety of existing drugs and new drugs. We're going to target the immune system, not give systemic stimulation of the immune system, like the case with the current immunotherapy, which is a nice advances, but still suboptimal. And we are going to gene therapy for limited mutation, like sickle cell, with the nanotechnology and with active targeting is going to become a reality, because we're going to improve the safety and efficacy. Biosensors, auto medicine delivery together, biocompatible, tissue engineering. We are going to actually, diseases are going to be subclassified into many different subtypes, because of the genetic information is going to enable us to, instead of putting, say, hypertension into hypertension, you're going to have hundreds subtypes of hypertension, to the point that each subclass will become an orphan disease. So this is what we think about the future. I want to thank you for your attention, and thanks so much.
Thank you, Professor Mousa, for this nice, informative, futuristic talk that can revolutionize our life, not only medicine. We can have one or two questions for Professor Mousa. Yes, Professor Maha.
Diabetes. You said that there is going to be an outbreak in insulin or oral insulin.
Yeah, oral insulin in a way is, there are a lot of attempts. Inhalation insulin was developed by Pfizer and failed after its approval. Anyway, the route of administration for insulin is the oral, because insulin actually oral is better than insulin injected. Why I'm saying this? Because the first pass when you take anything oral, it has to go via the hepatobiliary system. So it will land into the pancreas, very efficient delivery to the pancreas. Insulin, if you give it, it's a dose dependent. Insulin has a pro-inflammatory effect and anti-inflammatory effect. If you give it through the pancreas, it is an anti-inflammatory, and it's of course will bring the hemostasis of glucose. This is with the nanotechnology and with some targetability, it's actually is going to happen.
Any other
All right.
questions? Thank you, Professor Mousa. Thanks.